Okay, guys, I want to finish up the Chapter 9 notes today, and this is about where we ended. We were doing RNA processing, where the three steps involved are the introns are going to get cut out. And remember, there's like a base pair sequence here, like a something, I don't know, and a base pair sequence at the beginning and the end of an intron that will be recognized by bases in the, remember the SNRPs are made of RNA as well. And somebody was asking me, SNRPs, we write like that, sometimes we write like that, and the spliceosome is sort of the same thing. So once the SNRPs come together, they form kind of a complex called the spliceosome that cuts the intron out and then splices, puts together the exons. Okay, so we used to think that that the introns, once they got cut out, we used to think they were, junk DNA or they just got degraded. But now we think that even this little lariat, they call it, which is the intron cutout, might have some kind of function. We don't know what. I highly suggest watching the video link there at the bottom because it shows it kind of in motion. It's a good way to just review. And then here's what I was just saying, that the old view was that the introns were junk, but now we think they do something. They may be these small nuclear RNAs, these new RNAs that we said have been discovered in the nucleus, maybe introns that have been cut out, because remember, it'll just be RNA. Excuse me, wow. I cough. All right. Prokaryotes versus eukaryotes. I already kind of talked about that, but this kind of sums it up again for you. Most of what we talked about was eukaryotes, transcription, RNA processing. We just talked about that. And then we're going to do translation. And remember, only 2% of the DNA actually codes for proteins in a eukaryote. And so there's a picture that goes with that. And remember, in prokaryotes, they do transcription and then immediately do translation. There's no RNA processing. There's no introns. And they're kind of the opposite. In a prokaryotic cell, in a bacteria, 98% of their DNA, and they only have, remember, that one nucleoid region of circular DNA, 98% of theirs is actually coding for proteins. So they don't have as much non-coding DNA. They probably don't have to have as many regulatory areas of the DNA because, like I mentioned in class, they're just one cell. They don't have different tissue types like muscle, nerve, like we do. They don't have different times in development like embryonic development, puberty, etc. And again, that video link at the bottom shows kind of this in motion and this in motion. Again, a great review. Then in your notes, there's a place for you to, after watching the video or reading the notes, to read through these and see if you can guess, is it a prokaryote or a eukaryote, just kind of a testing of your understanding. So if you want to pause right now, you can try that. And then I'm going to show the answers here in a second. And you can just pause. Here's the answers to those. Again, you can pause and write that down so you can use it to study. But I am going to move on. All right. So again, reviewing the genetic code a little bit. Remember, everything's in kind of groups of three, like three-letter words. A group of 3 nucleotides in the DNA is called a triplet. And then once we base pair that into the messenger RNA, we call it a codon. And each codon in the messenger RNA is going to code for 1 amino acid in the protein. So in theory, if you have 4 codons, you're going to have a protein with 4 amino acids, except I mentioned in class really quickly at the end, if this was a full gene, and this is just showing like a little slice of one, there's going to be a stop codon at the end, and that doesn't code for an amino acid. So in reality, if there are if there were, let's say, 40 codons, there would only be 39 amino acids because the last codon is the stop. Okay. So it asks you here, just to see if you understand, like looking at the messenger RNA in red, how many codons are in this messenger RNA right here? There's 4, okay? How many nucleotides is that? Well, that's 12. So be careful what the question is asking. Each codon is 3 nucleotides. And in theory, this will code for 4 amino acids, but, not in a full gene. Not in a full gene. It'll be 1 less because of the stop codon. And the stop codon at the end doesn't code for an amino acid. The start codon does, which we're going to see here. Okay. Just a reminder, there's the 20 different amino acids that we can use to make proteins. But we say that the genetic code is redundant because there are 64 different combinations of codons. So you can do the math. We have 4 letters, and we're making 3-letter words. So there's 64 different combinations, which you can see in this chart here. But there's 64 codons, but there's only 20 amino acids for them to code for. And so what that means is some amino acids have more than one code. And why and how, and we don't really know. We just know this is what it is. But you'll see, for example, like leucine here, or let's do like valine. Let me skip to the next slide, which has kind of the answers here. Valine, if you read GUU, okay, so you just look up the first codon there, the second codon on the top, and the first nucleotide on the left, the second one on the top, and you see that kind of intersects kind of in this box down here, and then you look up the third one. So if we're doing GUU, that intersects right there, and you get valine. But you'll notice for GUU, GUC, GUA, and GUG, all 4 code for valine. And so often it's the last, we call it the wobble effect, but it's the last letter of a codon that doesn't matter. So if there's a mutation in the last nucleotide of a codon, sometimes it's okay. Sometimes it's the same amino acid. And we're going to call that a silent mutation at the end. So if you had a mutation that switched GUU to GUC, you're still going to get valine put in that place in the protein, and the protein is going to be fine. So it's silent mutation because it's not causing any difference in the protein. So just make sure you know how to use the chart. Okay. And the other thing I should point out, this chart, it'll be provided for you. You don't have to memorize it. But notice that this chart is messenger RNA. Okay. The big mistake most people make is they look up the DNA triplet or the anticodon on the bottom of the tRNA. So make sure you're looking up the messenger RNA codon, okay? If you're looking up the DNA, you'd probably figure it out pretty quickly, especially if it had a T in it, and you'd look at this and go, wait, there's U's. Oh, yeah, I have to look up not the DNA, but the RNA. So the chart just happens to be in messenger RNA language. So the other thing you noticed about the codons, there's one start codon. It's always AUG, okay? AUG is the first codon in every single messenger RNA, every single protein, okay? And it actually codes for the amino acid methionine. So methionine is the first amino acid in every protein. Sometimes it's then cut off later, but that's, kind of the same in every single protein. Then there are 3 stop codons that could be at the very last codon, and they don't code for an amino acid. And we're going to see that's why the ribosome kind of lets go at the end, okay? Read through this just about the genetic code being highly conserved. I told you this theme comes up a lot. Whenever we say something is conserved, it means it's the same in all life on Earth, and it shows that we're all related. And we're going to see this is really important. Like when we get into genetic engineering, I can take DNA from a frog and put it in a worm if I want to, for some reason, and the code is read the same, okay? So the genetic code, AUG is methionine in every single living organism on Earth that is doing translation. All right, so this is what I was just talking about. The wobble hypothesis is just saying that the last codon is sometimes the least significant, and if you had to have a mutation in either the first, second, or third, you should pick the third, okay? That's what we call the wobble, meaning like the last letter can sometimes wobble or change, and it doesn't affect the amino acid, okay? Not always. I mean, sometimes it does, but a lot of times it doesn't. So translation, let's get into translation, which is going to take place in the cytoplasm. So the messenger RNA has been processed and it comes out, and the ribosome is going to bind on and read it, okay? And so out here in translation, we're going to have all 3 RNAs involved. Messenger RNA is going to be read by the ribosome. Remember, the ribosome is made of RNA. Which I should mention, this comes up later, but do you remember when we learned the nucleolus and we said it has the DNA for ribosomes? So the genes for our RNA are actually in that region that, remember when we did the nucleus and it would kind of stain darker? For whatever reason, it stains darker, but that's the DNA that codes for ribosomal RNA. And then we're going to have tRNAs. So let's look at a tRNA, and you have lots of different tRNAs. They're specific ones in the cytoplasm, and what's important, this is really important, I mean, I bolded it and underlined it, but each tRNA will bind a specific amino acid. There's a binding site kind of up here on the top that fits a specific amino acid. This particular tRNA will only bind with arginine at the top, and at the bottom, it will have the specific anticodon that is going to kind of pair with the codon that means arginine, okay? So it's important, if tRNAs just picked up any old amino acid, then when they came into the ribosome, we would just get a random amino acid order. We wouldn't get a specific protein. Sorry. All right, so there are hydrogen bonds. Remember, this is just a strand of RNA, so like really, like there might be an A there and a U there that paired up, and maybe a G and a C, and oops, that's supposed to be a C. You know, maybe C, G, and then maybe U, A. And so this is a single strand of RNA nucleotides, but then it folds on itself, and it base pairs and forms hydrogen bonds kind of to form this cloverleaf shape. Okay, and then the 3 nucleotides that just happen to be at the bottom, we have to draw them really big like this. They're not like these really big letters sticking off of it, but they just make them big because these 3 nucleotides of this entire chain are important because they're going to pair with the codon in the messenger RNA, okay, which you'll see in the process here. These are just some different ways. You'll see tRNA in a picture. You know, it's a cloverleaf shape, so you'll see it, but this is more kind of what it really looks like. But notice at the bottom, they all have an anticodon, but you'll see them depicted sort of differently in different sources. Okay, so I think I already talked about that. You guys can read through all of this. It's all there for you to read. So this is my messenger RNA. And remember now, I have exons only because the introns have been cut out. And this picture doesn't show, but at the 5' end here, I have my MG cap, my methylated guanine that's at the 5' end. And remember, this is the beginning of the gene now in RNA. And then, you know, I have a poly A tail on the back here that it's just not showing. And then notice also, let me highlight it. This part that I'm highlighting is actually the gene, okay? But notice there's what we call, there's a leader sequence, which is just not coding for the protein, but just there's a little bit of bases before the gene, before the start codon, and there's often some bases kind of after the stop codon, and then the poly A tail. But this is, you see, here's my start codon, so that will be my first amino acid, AUG, and then here's my stop codon, okay? And we'll get into this more in chapter 10, but this leader sequence, we'll see this is important because I can block, like if I put something on here, let me do a different color. If I put something on here, you know, that blocks the leader sequence, that's going to prevent the ribosome from attaching. So another way we can control which genes are expressed into proteins, we can control it here, where we make the messenger RNA ahead of time and have it kind of ready and waiting. And then when we need this protein, or then we have an inhibitor that is supposed to bind to the leader sequence so that the ribosome can't read it, but it's sitting there ready to go. And then if I need this protein, then we will have something that removes this inhibitor, and then the ribosome can read it and make the protein. So that's another way to control when genes are getting expressed. It can be controlled at the translation stage here. Again, so all of those are filled in for you. You can read them. I don't know if this one is. So what would be the anticodon on the first tRNA that would bind the start codon? Okay, so if you look up here, if it's AUG, that's the first codon. The tRNA that's going to come in has to have the right base pairs to go with AUG. So the anticodon would have UAC. Okay, whoops. Okay, so you would have a tRNA like this that would have UAC on the bottom, and it would be bringing with it methionine on the top. Okay. The ribosome, I had you draw a picture of it the other night, but the ribosome itself, like when we draw a little dot like that, when we learned about cells, we drew ribosomes like a dot, but this is looking at it kind of, up close. It's actually made of two subunits, a large and a small, and then once they come together, you see there's kind of three kind of sites in there called the E, P, and the A site, where tRNAs can come in. And so the messenger RNA is going to feed through here, and the tRNAs are going to be coming into these sites. So I like to think of the A as the kind of the adding site, because this is where a tRNA will come in. Okay, so tRNAs will enter in the A site. And then the ribosome is going to slide down, and then eventually tRNAs, think of the E site as meaning exit. So the tRNAs will then leave the ribosome from the E site. They come into the A, and then they leave the E. Let's see. I kind of already talked about that. Again, another video, really good. I'm not going to, maybe I could get it to show. But it shows this whole process in motion here. And then I gave you kind of pictures in the notes as well. But what you'll notice is, so we have initiation, elongation, and termination. Again, kind of the same three stages we had in transcription. But notice initiation, you see the small ribosomal subunit binds to the messenger RNA first. It kind of attaches to the 5' end where the leader sequence is, all right? And then the start codon is sort of in the middle there. It's going to be in the P site. And then your first tRNA actually comes in before the ribosome is complete. So you'll notice my first tRNA has UAC, the anticodon, that goes with AUG, the codon, and it's bringing with it methionine, okay? And then the large subunit hooks on while that first tRNA is in the P site, and we're ready to roll, okay? So that's initiation. Now notice this picture that I happen to use only shows a P and an A site. There's actually an E site here. So just keep that in mind as you look through this. Then elongation is just going to be as the ribosome reads down, it's going to read down, the ribosome is going kind of in this direction. And, but first you're going to have your second tRNA come in. And notice if my codon is GUA, I have the CAU anticodon, and it's going to be bringing with it valine. And so if you look up GUA in the codon chart, it should say valine, because that's what GUA is coding for. It says we want to have valine as the second amino acid. And the way this works is that the tRNA that has CAU will only have a binding site that fits valine, okay? So that's going to come in, and then you're going to have two tRNAs that are in there at the same time, and a peptide bond is going to be formed between the first and the second. So here's the beginning of our protein, and then it's just going to slide down. The ribosome is just going to slide down one codon, and when it does, it slides down one, the first tRNA that brought methionine, he's done, and he's going to leave. Okay, and remember, he's really leaving the E site, but and so he can go out and look for another methionine, and maybe we need him later. Okay, so it's going to slide down one, which means that this one that was in the A site now goes to the P site. Okay, so this was our second tRNA, and then a third one's going to come into the A site, and again, codon pairing with anticodon, bringing the right amino acid, and that's going to come in. And so you always have sort of the 2 in the P and the A site, and then you're going to form another peptide bond between valine and arginine, and it's going to just keep doing that. It's going to slide down one codon, a new tRNA, the old one leaves, and that's elongation. And eventually you're going to get, you start getting, here's our protein or a polypeptide chain sticking out of the top of the ribosome, and that's hopefully the right order that we wanted. And when you get to the stop codon, this is termination, UGA, there is not a tRNA that goes with the stop codons. There's something that's called a release factor that will come in and bind, and that just basically pries everything open. So the ribosome kind of comes apart, lets go, the messenger RNA is free, and it's done. Okay, so that's translation, essentially. And remember, that's 3 RNAs. I have my messenger RNA here being read. The ribosome is made of rRNA, and I have the tRNAs bringing in the amino acids. And again, these videos are really good. I'm not going to bore you with it here. You can watch them on your own. I highly suggest that. Okay. And then I think, well, those are in your homework. I'm not going to go over the answers to those because you're going to do it. So that is it. Okay. So that's translation. And so we went through the whole process: transcription, RNA processing, and translation. And that is how you make a protein. Now, I did want to talk about one more thing. Sorry. Psych. Mutations then, all right? So a mutation is when there's a mistake in this code. And a point mutation is the word they use for like a small magnitude, like one or a few nucleotides. Different from, like, remember when we talked about karyotypes, you might be missing a whole chromosome or a whole arm of a chromosome, or those would be like bigger mutations. Okay, so a point mutation is just one or a few letters, nucleotides, in one gene. So we're just talking about, a mistake in just sort of one gene here. And there's different kinds. There's substitution, which is exactly what it sounds like. Like one nucleotide might get switched for another. You're supposed to have an A and a T gets put in your DNA or something. And sometimes that can be silent, like I showed you before, where you might get the same amino acid. So I forget the example, but like I forget the valines examples, but I don't want to go back. But let's say what was the valine ones? I don't have it. I think it was like, here it is, like GUU might get switched to GUA. Okay? And those are both valine. They both code for valine. And so that would be a silent mutation. You'd have no effect. A missense mutation is one in which you might switch a letter in a codon, but one amino acid gets replaced with another. Again, keeping in mind, that's only one amino acid in a large protein that could be thousands of amino acids long. And so sometimes just changing one amino acid in a big protein, the protein might still fold up in its shape correctly and be fine. It just depends sort of on where that amino acid is. And then a nonsense mutation is when you have a substitution that you end up getting a stop codon kind of in the middle of the gene somewhere. So if you had, like UAC, an example of this would be like UAC, which is supposed to code for tyrosine. And if that gets switched to UAUG, that's a stop codon. So as soon as the ribosome gets to the stop codon, it's going to let go, and it won't read the rest of the gene. So that could be really bad. So, if you had to pick one of these, which one's worse, again, it all depends. But if I had to pick one of these, silent mutation is the best. These two, I would say the nonsense is the worst one if you had to pick one. Then deletion and addition are also what we call frame shift. It's a big word they talk about. So you either delete or add an extra nucleotide, gets added in there somehow. And remember, we read everything in sort of groups of 3. And so if you add an extra base in there or you take one out, it slides the reading frame. And everything from that point on will be wrong. So I like to use the example of, like, let's suppose I had a sentence like, the cat ate the rat. So it's a sentence that's got all 3-letter words, kind of like we would read DNA and RNA and codons. And if I take out the A, and again, these spaces don't exist, right? And but the first letter or the first word would still be the, but the next 3 letters would be CTA, and then the next 3 letters would be TET, and then you see how from that point on, where I deleted that base, from that point on, I have all different 3-letter words, okay? And so that's what we call a frame shift. Same thing would happen if I added a letter in. So if I have the cat ate the rat and I added an extra letter in here, it's still going to get read in groups of 3 no matter what. It's going to shift the reading frame. You're going to get different amino acids from that point on. So deletion and addition are better if they happen maybe toward the end of the gene, because the gene would be fine, and then maybe the last few amino acids are, switched out from the frame shift. That's a deletion, addition, and frame shift are often the same thing. This is just an example of a substitution missense, okay, where one amino acid gets switched out for another, and this is the one that causes sickle cell anemia. So the gene that codes for hemoglobin, if you remember, hemoglobin is the protein that is in red blood cells, okay, that carries oxygen for us, okay, and people with sickle cell anemia have only this one little substitution. So the DNA is supposed to be GAG, but this A gets switched, this one base pair gets switched, okay? And so you notice the RNA, which is supposed to be GAG and code for glutamic acid, that codon says GUG, which codes for valine. So valine gets put into one place in the hemoglobin protein instead of glutamic acid. And this is an example where just switching that one nucleotide substitution switches one amino acid in a very large protein, but it happens to have a big effect on hemoglobin and how it forms its quaternary structure. Okay, so that's just a picture of hemoglobin. And so what happens is normal hemoglobin tends to be sort of like globular in shape, and the sickle cell hemoglobin, that valine, for whatever reason, causes them to clump together and stick together. And so inside of a red blood cell, you get sort of these, this kind of leads to your remember, hemoglobin is inside red blood cells. It makes the red blood cells kind of these long sickle shapes. And I don't know if you know, a sickle is like one of those giant, it looks like a machete that you would chop down crops with, that has this shape. Okay, and this is a problem because these are going to get stuck in small capillaries and things like that. And so that's just one example. This is showing you a picture of a nonsense mutation where, this is your normal sequence here, and you're supposed to have, this, and then if you suddenly get a stop codon, that's interesting that has the same amino acid a bunch in a row. But anyway, if you switch this one letter here and you end up with a stop codon, like the rest of this protein won't get made. That's probably going to be bad. This is just showing you a frame shift, kind of like I did with the cat ate the rat, but just kind of look at it and you can see, here, this shows you an insertion and how the reading frame gets shifted. Okay, so I think that's it. All right, so those are mutations, and you have some of those at the end of your worksheet. All right, so now I am